Alright, these next series of videos are going to focus on the MHC, or Major Histocompatibility Complex. And everything I'm going to talk about in this next set of videos are covered in your textbook on page 145 to 148 and pages 135 to 136. And I have to say that these next sets of videos are probably the most confusing topic that we're going to cover in this course. So a lot of stuff is going to go on, but hopefully you get a chance to review it, ask questions during class, look in your textbook if you don't understand it. But the MHC is very important to understand if you would like to understand how T cells work, how the body can defend against so many different infections, and as I mentioned in class, it has to do with organ transplantation. So that term histocompatibility, MHCs were identified as regions of the genome that were required or important for tissue donation, organ donation. So the MHC, or Major Histocompatibility Complex, it actually refers to the genes for the molecules that we've been talking about. So we talk about the MHC I or MHC II molecules. MHC actually refers to the genes, the area of the genome on chromosome 6 is this huge area on chromosome 6 that encodes for lots of genes that have to do with compatibility of organ transplantation. But it turns out that these genes actually make proteins that are involved in antigen presentation. So MHC technically refers to the complex on the genome, but we also use it when we talk about the proteins that are found on the surface of the cells that are involved in antigen presentation. So let's start with chromosome 6. And so here's chromosome 6. You have two copies of chromosome 6, one from mom, one from dad, right? And so there's a gene. There are genes on chromosome 6 that encode for the protein that makes up the MHC class I molecule, right? Now, the other term that you're going to see us using throughout this, throughout these videos and in your textbook is HLA. Which stands for human leukocyte antigen. So that's another term that people use for the protein found on the surface of cells that are involved in antigen presentation. So MHC class I and HLA class I, they mean the same thing. They refer to the proteins on the surface of cells. That being said, we're going to start off with the HLA-A gene. So that's the name of the gene, HLA-A, right? And that gene encodes for the protein that is the alpha chain of the MHC class I molecule, or the alpha chain of the HLA class I molecule. Again, it really doesn't matter which term you use because they're both really the same thing. But here's the gene, HLA-A. It's a gene, encodes for protein, and there's the protein. Remember, the alpha chain is the one that presents peptide. That other chain is the beta-2 microglobulin that's encoded by the beta-2 microglobulin gene, which we're not going to worry about. We're going to focus on the genes that encode for the alpha chain protein of the MHC class I, or now I'm going to say the HLA class I. Now, from genetics, you know that sometimes genes have a number of different alleles. So they can have 2 alleles, they can have 3 alleles. In this instance, we're not going to use the term alleles, and your textbook uses the term allotypes, which is basically another way to talk about alleles, different forms of the gene or different versions of the gene. And so in your textbook, it says that there are 506 different alleles or different allotypes of the HLA-A gene. Why so many different allotypes? Well, that's a very good question, and we're going to have to address that, right? So let's give an example. Let's talk about 2 different versions or 2 different allotypes of the HLA-A gene. What's different about them? So we'll start with version 0201, and the nomenclature is always kind of weird, right? But that's the real version of the HLA-A gene, right? And that's going to make a protein, which we'll refer to as the HLA-A 0201 isoform. So here's another new term, isoform, which can refer to these different versions of the protein that are found on the cell surface. Right? And let's say the other HLA-A allele is 3001, and that's a real HLA-A allele. It's a real piece of DNA. It's been sequenced. We know the DNA sequence of that allele. And that's going to make a different isoform, the HLA-A3001 isoform. Now these are both MHC class 1 molecules. They both present peptides, but there's something different about them. So let's examine the first one. Let's examine 0201 first off. So next slide, right? And this is a topic that is actually covered in your book on page 135 that has to do with the peptide binding groove on the MHC class 1 alpha chain protein. So we drew a little close-up here of MHC1. There's the alpha protein and the beta-2 microglobulin. Right? And so I've just been saying, well, peptides get loaded in here and presented to CD8 cells. Well, what peptides exactly can fit in this? Can every single peptide fit in there? No, actually, quite the opposite. Very specific peptides can fit in there. Typically, peptides range only from the size of 8 to 10 amino acids long can fit in this peptide binding groove. So longer peptides, shorter peptides typically don't get in here. So if a peptide is anywhere from 8 to 10 amino acids long, then it is possible to get loaded in here, but it's not definitely going to get loaded because there's some other constraint for MHC peptide loading. So let's look at this example. Let's look at this peptide. It's a 9-mer, right? 9 amino acids long. And so what determines which peptides can load into an MHC1 are the amino acids that are encoded for in the protein. So the amino acids in the peptide binding groove interact with specific amino acids in the peptide. So if we'll start off with allotype A0201, that allele makes this protein with certain amino acids, and those amino acids are going to help the peptide, not every peptide, but only peptides with specific amino acids load into here. And so we're going to talk about anchor residues. So typically at amino acids 2 and 9, that's where the major contact is between the MHC alpha chain and the peptide. So there are specific amino acids in the MHC molecule that interact with anchor residues in the peptide. And typically the anchor residues reside at amino acids 2 and amino acid 9. So that means allotype A0201 can primarily bind only peptides that have a leucine at amino acids 2 and a valine at amino acid 9. So the peptide I'm showing here, it will fit very nicely in this peptide binding groove. And this peptide can get loaded here onto the MHC class 1, allotype A0201, and that peptide can be presented to a CD8 positive T cell to see if that T cell has a T cell receptor with a variable region that binds and interacts with this peptide. Right? So this is the allotype HLA-0201. So when we talk about its what peptides it can present, we usually draw something like this, where basically there's a leucine, position 2, and a valine in position 9. So any amino acids, practically any amino acid that looks like this, can be presented. So anything, the amino acid in position 1 and amino acids between positions 3 to 8, those pretty much can be anything. So this peptide can be presented, the one on the top can be presented, the one on the bottom can be presented. So there is some promiscuity. So we talk about the promiscuous binding site of MHC molecules. This won't present every single 9 amino acid peptides, but it will present every single 9 amino acid peptides that have the anchor residues that interact with the amino acids in the MHC1 allotype 0201. So I'm going to stop the video here. The next part we'll talk about peptide loading, and we'll continue with this discussion.